Welcome back to the deep dive. So today we're really opening up the hood on life itself. We're talking about the human genome.
We are, but we're going way past, the biology 101 stuff.
Right. This isn't about dominant and recessive genes. We're looking at a landmark paper from Google DeepMind that, well, it basically proposes a new operating system for understanding our DNA.
It really is a shift in perspective. Everyone knows the central dogma, right? It's sort of the golden rule of biology.
DNA makes RNA makes protein.
Exactly. It's clean, it's linear, it's in every single textbook.
It's almost like a commandment.
Yeah.
The scripture of life.
Right. But here's the thing that keeps computational biologists up at night, that central dogma, it really only accounts for about, 2% of your DNA?
Just the protein coding stuff.
Just that, which leaves the other 98%.
The famous junk DNA.
Exactly, though we really can't call it that anymore. That 98%, the non-coding genome, it's not junk. It's the management.
It's the logic gates, the switches, the dimmers that decide when and how those proteins get made.
And until very recently, trying to decode that massive operating system with AI has been, a nightmare of trade-offs.
And that is the perfect setup for the paper we're breaking down today. We are looking at this new model called AlphaGenome. And the core promise here seems to be about resolving a paradox.
It is.
From what I gathered, before AlphaGenome, if you wanted to analyze DNA with a computer, you had to make a painful choice.
You did. You could have a wide field of view or you could have high resolution. You just couldn't have both.
That's the resolution versus context dilemma.
That's it, exactly. Think of it like photography. You have these models, like Informer or Borzoi. They're like wide-angle lenses.
Okay.
They can look at huge stretches of DNA, which is crucial because biological instructions can be spread really far apart. But the image they get it's blurry.
Yeah.
They group things into buckets of 128 base pairs.
So they see the forest, but they completely miss the individual trees.
Precisely. On the other hand, you have specialist models like Splice AI. They have a microscope.
Okay, super high res.
Perfect single base pair resolution. They can see every single letter, but they can only see a tiny, tiny sliver of DNA at a time.
So they see the veins on a single leaf,
Yeah.
but they have absolutely no idea what forest they're even standing in.
And biology just doesn't work in isolation, A gene over here might be controlled by a switch 100,000 letters away.
If you're using the microscope, you miss the switch.
And if you're using the wide-angle lens, you miss the precise mutation that broke that switch.
Enter AlphaGenome. The mission here is to break that trade-off, to build a generalist model.
And the specs are they're impressive. Takes a massive input.
Features at single base pair resolution.
It's the best of both worlds.
I want to pause on that number, though. 1 million base pairs. Why that specific number? Is that just a nice round figure, or is there a biological reason for it?
Oh, it's very intentional. The paper notes that 99% of all enhancer gene pairs, so basically the relationships between a gene and its control switch,
Right.
they fall within a distance of 1 million base pairs. If your window is smaller than that, you are biologically blind to the connection. You just can't see it.
So they built a window big enough to see the whole picture. I really like the analogy they used in the paper. It's like reading a novel.
It's a great way to visualize it.
Previous models could either, summarize the plot of the whole book but miss the individual words.
Or they could spell check a single sentence, but have no idea who the main character even was. AlphaGenome reads the whole novel and analyzes the inconsistency of a single period.
That is a very bold claim.
Yeah.
So how on earth did they actually build this thing? Let's get into the engine room. Section 1. Architecture and training.
Right.
This isn't just a standard neural network, is it?
No, not at all. It's a Frankenstein's monster, really, in the best possible way. It's a hybrid architecture.
Okay, so they mashed up a few different things.
Exactly. They took two very different types of AI technology. First, they used a UNet backbone.
UNets. I know those. Those are usually for image processing, right? Like looking at medical x-rays.
That's the one. UNets are brilliant at handling structure. They have this kind of hourglass shape. They downsample the data so they compress it to understand the gist, the high-level features.
And then they upsample it back out to restore all the detail.
Yes. So the UNet is the part that allows it to sort of zoom in and out. It handles the local structure. But right in the middle of that hourglass, at the most compressed point, they inserted a transformer tower.
Transformers. Okay, so that's the tech behind LLMs like ChatGPT.
Yes. And the reason it's there is very specific. Transformers are the kings of attention. They can look at a token at the very beginning of a sequence and relate it to a token at the very end.
Okay, so in this context, the transformer is what connects the switch at base pair number 1 to the gene at base pair, like 900,000.
Spot on. The UNet handles the local texture and grammar of the DNA, and the transformer handles the long-range plot points.
And doing this is not cheap, computationally. I saw a note in the engineering section. They had to split that 1 million base pair sequence into chunks across 8 distinct TPU devices just to handle the math.
Oh yeah, that's sequence parallelism. It's a massive engineering feat just to get the data to fit in memory. But honestly, the training strategy is even cooler than the architecture.
You mean the teacher and student approach?
The very one. It's called distillation.
Okay, I find this concept so fascinating. Walk us through it.
So training a model this complex on raw data is incredibly slow and heavy. So first, they trained these massive teacher models on all the available data accessibility, expression, splicing, you name it.
So these teachers are super accurate, but they're these huge, unwieldy beasts.
Exactly. The wise old professors who know everything but move very, very slowly. Then they initialize a single student model. The student's only job is to mimic the output of the teachers. But here's the twist. They train the student on randomly augmented sequences. They would shift the DNA slightly or flip it to its reverse complement.
Wait, so they're feeding the student distorted, messy data on purpose?
Intentionally. They are throwing curveballs at it. It's like learning to drive in a simulator where the road keeps shifting or the mirror suddenly flips upside down.
And by forcing the student to predict the teacher's answer, even when the input is messy,
the student actually becomes more robust than the teachers were.
That is wild. So you end up with a student model that is faster, lighter, and tougher than the originals it learned from.
And the result is a single model that runs in under a second on a GPU, but matches the accuracy of that massive committee of teacher models.
Okay, before we move on to the actual biology, let's do a quick recap of the tech here, because this is really the foundation for everything else.
Good idea.
First, the concept. AlphaGenome breaks that trade-off. It gives us the wide angle and the microscope at the same time. Second, the stats. It takes in 1 million letters of DNA and gives output at 1 base pair resolution.
Then the tech. It uses that hybrid architecture, the UNets for local structure, and the transformers for the long-range connection.
And finally, the method. It uses distillation to compress all that knowledge into a single, fast, accurate student model. So a check-in for you listening. Can you visualize that? The UNet squeezing the data down, passing it to the transformer to connect the distant dots, and then expanding it back out to predict every single letter.
Because that really is the magic here. But, you know, a model is only as good as what it predicts, and this is where we get into the biological killer apps. And the first big leap forward in this paper is splicing.
Splicing. Okay, so this is the editing room of the cell.
That's a great way to put it. When your DNA is transcribed into RNA, it includes a lot of raw footage called introns. These need to be cut out, leaving only the exons, the good scenes, to be stitched together to make the final movie, or protein.
And if the editor falls asleep and leaves a bad scene in, or cuts a good scene out,
you get a non-functional protein, you get genetic disease. Now, previous models like Splice AI were good at finding the cut sites, but AlphaGenome goes further. It predicts the full trilogy.
Which is?
Splice site, so where to cut. Splice site usage, which is does the cell actually use the cut. And splice junctions, the physical connection itself.
I saw the paper mentions a specific mechanism for this, the splice junction head, that explicitly models that interaction. But let's look at a concrete case study from the source material to see why this matters. The DLG1 gene.
This is a perfect example of why that single base pair resolution is so critical. There is a specific variant in this gene, a tiny 4 base pair deletion. The sequence TACTC just becomes T.
That's it, a 4-letter typo.
A tiny typo. Previous wide-angle models wouldn't even see it. But AlphaGenome saw something very specific. It correctly predicted exon skipping.
Meaning the cellular machinery just skipped over a whole section of the gene.
Yes. Because that tiny deletion broke the signal for a slice site. AlphaGenome predicted the loss of usage at that site, and crucially, it predicted the appearance of a brand new junction that bypassed that exon completely.
It saw the missing scene in the movie.
It did, and there was another one, COL6A2, that went the other way, right?
Right. In that case, a single letter change, a G to a C, created a new spliced donor signal, an imposter signal.
And AlphaGenome modeled the competition between the old, correct signal and this new one. It predicted an extended exon, The machinery kept reading past the old stop sign to this new one.
The key takeaway here is that it beats the specialists at their own game. It outperformed tools like Splice AI, especially on those deep intronic variants.
Mutations that are far away from the obvious action but still manage to mess everything up.
Which brings us to the second major pillar here, gene expression. It's not just about how you edit the RNA, but how much of it you produce. The volume knob.
This is where we talk about EQTL's variants that change expression levels. And honestly, this has been a weak point for AI models for a long time. Most models can tell you something will change, but they are terrible at telling you the direction.
The sign prediction. Does the gene expression go UP or does it go down N?
It seems so simple, right? But biology is incredibly noisy. AlphaGenome improved this prediction metric significantly, from 0.75 to 0.80 ROC compared to the previous best model, Borzoi.
That sounds a little abstract, so let's ground it in an example. The APL4 gene, what happened now?
Okay, so here we have a variant where an A becomes a C. AlphaGenome looked at this and correctly predicted that this mutation, this single letter change, kills a splice donor motif.
So the splicing process fails.
It fails. And because the splicing fails, the RNA transcript becomes unstable and it just degrades. The model correctly predicted that expression would drop. It nailed both the direction down and the magnitude of that drop.
But this all links back to the architecture again, doesn't it? Because sometimes the thing controlling the gene isn't right next to it. It's that action at a distance you mentioned earlier.
Exactly. An enhancer is like a gas pedal for a gene. But in the genome, the gas pedal might be 100,000 base pairs away from the engine.
And because of that transformer tower we discussed.
AlphaGenome outperformed Borzoi in linking enhancers that are over 10 kilobases away. It can solve that 3D puzzle in a way that others couldn't.
It even figures out the tail of the RNA, this thing called polyadenylation, without being explicitly trained on it.
That was a surprise finding, even for them, I think. Just by learning RNA density, it learned to model the competition for where to cut the end of the RNA string. It's emergent behavior. The model just figured out the rules of the game by watching it being played enough times.
Okay, let's do another quick recap before we get to what is really the ultimate test case in this paper.
Sounds good.
So, on splicing, AlphaGenome models the full trilogy: sites, usage, and junctions. It catches complex errors like the exon skipping in DLG1.
For expression, it's significantly better at predicting the sign, whether a gene's activity goes up or down.
And for range, it successfully links those gas pedals, the enhancers, to the engines, the genes, across huge genomic distances. So a question for you listening. If a mutation happens 50,000 base pairs away from a gene, how does AlphaGenome know if it even matters?
The hint is to think about that transformer tower connecting page 1 to page 100.
Now we have to talk about the killer app case study.
Yeah.
This is where the rubber really meets the road. T-cell, acute lymphoblastic leukemia, or TAL,
A very serious cancer. And the villain here is an oncogene called TAL1.
which should be turned off in these cells.
It should be completely off. It's a dangerous gene. In leukemia, it gets turned on. And the researchers looked at a specific mutation, an insertion of just a few letters, located 7.5 kilobases away from the gene.
So not on the gene, but nearby. Let's walk through the in silico simulation. They feed this mutation into AlphaGenome. What does the model see?
It sees a whole sequence of events. First, right at the site of the mutation, it predicts a huge spike in activating histone marks, things like H3K27 Hec.
So it sees a neo-enhancer being born, a new gas pedal just appearing out of nowhere.
Exactly. Then, second, 7.5 kilobases away, at the TAL1 gene itself, it predicts a decrease in repressive marks. The brakes are being taken off the gene.
And third,
A massive upregulation of TAL1 RNA, and the gene roars to life.
but here's the part that really got me. The researchers didn't just take the model's word for it. They used something called ISM-N, silico mutagenesis, to ask the model why. Why did this happen?
Right, they systematically mutated every single letter in that region to see what the model was reacting to. And AlphaGenome pinpointed that the insertion created a binding motif for a transcription factor called MYB.
So the model reconstructed the entire crime scene.
The entire chain of causation. The mutation created a landing pad for MYB. MYB landed and created a new enhancer. That enhancer reached over 7,000 base pairs to the TAL1 gene, turned it on, and that causes cancer.
All predicted from sequence alone.
Yeah.
That is the holy grail of computational biology.
It really is. It connects that tiny mutation to the molecular consequence across a significant distance.
It's incredible. But I have to play devil's advocate here. We've seen hype cycles in biotech before. Why does this model work when others have failed? The paper includes these ablation studies, basically breaking the model on purpose to see what matters.
And they found 3 ingredients were absolutely non-negotiable. First, resolution.
The microscope view.
Right. Training at 1 base pair resolution was crucial for splicing and chromatin accessibility. If you tried to blur it to 128 base pairs like the older models, the performance just collapsed. You need to see the letters.
Yeah, the second.
Context. There is a chart in the paper that shows performance dropping in a straight line as you reduce the view from 1 million base pairs down to 32 kilobases. You simply cannot see the regulatory landscape through a keyhole. You need the panoramic view.
And the third one was multimodality.
And this is the most interesting one to me. Training the model on everything, accessibility, expression, splicing, all at once, helped it learn better representations than if it had just studied one thing in isolation.
It's like how learning to play the piano helps you understand music theory, which then helps you play the guitar better.
That's a perfect analogy. Learning how splicing works actually helped it understand gene expression better. It truly becomes a generalist.
So we have a model that is a generalist, it beats the specialists, and it unifies our view of the genome. Where does this go from here?
Well, the immediate future is in diagnostics. We all carry what are called variants of uncertain significance, or VUS.
Right, a doctor finds a mutation in your genome, but has no idea if it's benign or if it's the cause of a disease.
And right now, figuring that out requires a wet lab experiment that could take months or even years. AlphaGenome acts as a virtual lab. You could run it in seconds.
And it opens doors for synthetic biology, too. Designing RNA therapies or synthetic enhancers.
Exactly. Things that only turn on in specific tissues.
But I want to end on a provocative thought they mentioned in the paper's conclusion. AlphaGenome is amazing at predicting molecular effects, what happens inside the cell. But it doesn't predict phenotype directly. Not yet.
Right. That's the last mile problem. It can tell you this gene's expression will drop by 50%, but it can't tell you you will get diabetes or you will be tall.
There's still a massive gap between the cellular effect and the whole body effect.
It's the difference between knowing the engine is running hot and knowing if the car will actually win the race.
And that gap will take more than just better algorithms, I imagine.
It will take massive amounts of clinical data to close, but AlphaGenome has built the bridge halfway across that river.
Well, we've covered a lot of ground today, from the UNET backbone all the way to the Tau1 oncogene. It really feels like we're entering a new era here.
It's a glimpse into a future where biology is as much a computational science as it is a wet lab one.
So we want to hear from you. If you could run just one mutation through AlphaGenome, maybe something that runs in your family, or just a scientific curiosity, which gene would you target and why?
I'd be fascinated to see what people would want to look for.
Let us know. Thanks for diving deep with us.
Until next time.